we have discussed how the anteroposterior segmentation genes subdivide the embryo into different segments by providing a coordinate system where a cell can determine its position by reading which particular segmentation genes, the maternal ones like Bicoid, the gap genes like cripple, payroll genes like even skipped, and segment polarity genes like hedgehog, which combination of these genes are expressed. The cell, by reading this combination, is able to tell its position. But how does a cell read the expression of these genes? It turns out that these genes are in fact transcription factors, just like Hox genes are transcription factors, and they bind sequences, DNA sequences called binding sites, which are part of stretches of DNA called cis-acting regulatory elements or enhancers, and cis-acting regulatory elements are the same synonym for enhancers. And in eukaryotic gene regulation, when transcription factors bind enhancers, they recruit RNA polymerase at the promoter and cause transcription, or alternatively, if it's a repressor that binds to the cis-acting regulatory element or enhancer, it can inhibit the RNA polymerase from binding to the promoter, and therefore it inhibits transcription. And one can analyze the expression pattern, or the role these transcription factors play in regulating genes, and thereby providing information to cells through an experiment known as a reporter assay. And what you do in a reporter assay is you build a synthetic gene where you have a reporter gene whose RNA or protein can be detected easily using in situ hybridization or immunolocalization, respectively. And this synthetic gene also has the promoter, and you can introduce different enhancers here, for example, A, B, or C, into the synthetic gene, making 3 different versions of the synthetic gene. This synthetic gene is introduced into embryos, and after several steps, you can carry out experiments to detect the RNA or protein expression from the reporter to know where a particular enhancer causes, when and where a particular enhancer causes genes, this reporter gene, to be expressed. The fundamental question to answer about how segmentation genes provide this coordinate system for the embryo is, how do you get this progressive refinement where Bicoid is expressed in this gradient from the anterior to posterior, so it goes down gradually, and then gap genes like hunchback are expressed in broad domains. In the case of hunchback, it's expressed only in the anterior half of the embryo, and then there is further refinement into payroll genes where they're expressed in narrow stripes, and so on and so forth. So the main question we have is, how do you go from something that's gradually changing into something that's more and more restricted or expressed in narrower and narrower regions of the embryo? So the first sort of answer to this question was provided by mutant analyses, where you took embryos that were mutant for Bicoid, for example, and looked at how the other segmentation genes are expressed in these mutant embryos. As it turns out, if you have an embryo that's mutant for Bicoid, this whole anterior expression of hunchback is missing. And what that tells us is that Bicoid must be an activator of hunchback, which means if we have a promoter for the hunchback gene, and here's the promoter, there must be enhancers that the Bicoid protein binds to and then recruits RNA polymerase to the hunchback promoter to drive transcription. So that tells us that the expression of segmentation genes is not independent of each other, but is in fact that they regulate each other's expression. The second part of the question is, how do you go from these gradients to something that is expressed in a restricted part of the embryo? And this question was answered by the sort of reporter experiments that we discussed just a minute ago. And so first, the experiment was that you had a reporter gene and a promoter, and several different synthetic genes were built. The first gene here at the bottom contained no enhancer, so it did not contain any Bicoid binding sites. The second version of the synthetic gene contained an enhancer that had only one Bicoid binding site. The third version had an enhancer with two Bicoid binding sites, and the third enhancer had three Bicoid binding sites. And when the expression of the reporter was measured in embryos, the synthetic gene that had no Bicoid binding site, no enhancer, had no expression. But as you increase the number of Bicoid binding sites, the expression of this reporter gene increased as well as extended further and further posterior into the embryo. And this pattern suggested that there is a certain minimum amount of Bicoid that's required for hunchback transcription to occur. And so when you have just one Bicoid binding site, you need a very large amount of Bicoid. And recall that there's more Bicoid in the anterior and less Bicoid in the posterior. You need a very large amount of Bicoid in order to recruit RNA polymerase and drive expression for hunchback. Once you have two Bicoid binding sites, then the minimum amount of Bicoid required is smaller, because there are more binding sites, more locations on the DNA for Bicoid to bind, and therefore there are more Bicoid molecules, more chances for Bicoid molecules to bind the DNA or stick to the DNA, even when you have a less amount of Bicoid. And finally, if you have even more binding sites for Bicoid, then the minimum amount of Bicoid required to drive transcription is even smaller, and therefore the expression extends further into the posterior. But the key point here is that if you have Bicoid higher than this minimum amount, higher than this threshold, then hunchback expression occurs. And if you have a Bicoid amount that's less than this minimum amount, then hunchback expression does not occur. And that is what explains the fact that even though there is a Bicoid present in sort of gradually decreasing amounts as you go from the anterior to posterior, hunchback expression is restricted only to the anterior half, and that's likely because the amount of Bicoid in the posterior half of the embryo is less than that minimum amount, that threshold of Bicoid protein concentration required to drive hunchback expression. And this also gives you a way to locate these borders, the region of the embryo where hunchback changes from on to off is called a border, to locate the borders of gene expression domains at different parts of the embryo. So if you want a gene to be expressed only in the head, then it would have only a small number of Bicoid binding sites. Whereas if you want the expression to extend further into the posterior, that target gene would have more Bicoid binding sites. Now, this threshold dependent activation of Bicoid target genes such as hunchback and all the other gap genes and pair rule genes are also targets of Bicoid, can explain how you can get localized expression where you have a expression in the anterior but not in the posterior. But how are we to explain expressions that are localized in stripes or domains, such as the expression of even skipped, which is expressed in 7 stripes, or the expression of crupple, which is expressed in a broad domain in the center of the embryo? Now, insight into how you can have two borders, one on the posterior side and the other on the anterior side, came through the analysis of the regulation of even skipped. And it turns out that even skipped, and this is the transcription start site, and this would be the promoter of even skipped, has several different enhancers, and each enhancer is dedicated to directing the expression of even skipped in particular stripes. So this blue one drives expression in this region of the embryo, which corresponds to the second stripe of the Eve expression. Another enhancer called the stripe 4-6 enhancer drives expression in the 4th and the 6th stripes, and the 3-7 enhancer drives expression in the 3rd and the 7th stripe, and then there is a 4th enhancer for the expression of even skipped in the 1st and the 5th stripes. Recall that enhancers are DNA sequences that contain binding sites or patterns for transcription factors to recognize and bind. And the stripe 2 enhancer contains the binding sites for several different transcription factors. There's Bicoid and also hunchback, and Bicoid and hunchback act as activators. That means they recruit RNA polymerase and increase the level of even skipped transcription. There are two other gap genes that regulate this stripe 2. There's binding sites for crupple, and crupple is a repressor, so it inhibits the recruitment of RNA polymerase, and as well as giant, which is also a repressor of stripe 2. So how can this organization of the stripe 2 enhancer result in a expression of even skipped in a narrow domain? So in order to understand this, we're going to draw the expression of these genes along the anteroposterior axis, and so left is anterior and the right is posterior, and on the y-axis we will show the expression level or the concentration of these proteins. Okay, and we have Bicoid, which is expressed like this in this gradual gradient. So in red we have Bicoid, and in orange we have hunchback, and so hunchback is expressed in the anterior half of the embryo, and it is not expressed in the posterior half of the embryo. Then you have the other two gap genes, the repressors, and we'll show giant in green. So here's giant. And then we have cripple, which we will show in blue. And so this blue we have cripple, and green we have giant, and these two are repressors, whereas in red and orange we have Bicoid and hunchback respectively, and these are activators. So if we look at how even skipped will be expressed only under the control of the activators, just like with hunchback, it will have high expression in the anterior and gradually reducing expression all the way to the posterior. And this is what happens when you have a gene that's activated by Bicoid, which is high in the anterior, and so it activates it in the anterior, and there is less Bicoid in the posterior, so there is less activation available in the posterior. Now, cripple, because it's expressed in the posterior, it will repress the expression of even skipped because there are cripple binding sites in the stripe 2 enhancer. And what that means is that the repression from cripple will prevent even skipped from being expressed in the posterior, and therefore you will get a sharp posterior border where to the anterior of this line even skipped is expressed, and to the posterior of this line even skipped is not expressed. However, even skipped is also repressed by giant, and giant is expressed to the anterior of the stripe, and so giant is going to repress it in the anterior, and therefore you will form an anterior border where to the posterior of this line even skipped is expressed, but to the anterior of this line even skipped is not expressed, since giant is expressed in large amounts in the anterior of the embryo and it represses even skipped. Therefore, through the combination of generalized activation, broad activation, but combined with localized repression, we are able to produce expression in a narrow stripe or domain of the embryo. And this is how progressive refinement is achieved, where there are activators that activate the gene in broad swaths of the embryo, but the enhancers contain binding sites for combinations of transcription factors that narrow down the expression through repression in the specific areas of the embryo. And therefore the body plan of Drosophila embryos and hence of the larvae and adults is specified by the combinatorial regulation of genes by transcription factors, and this combinations are encoded in the DNA in the form of the combinations of binding sites that are present in enhancers, and therefore during development, what is happening is that this combination, this code or the body plan is being read from the genome through the regulation and expression of these genes that get expressed in these progressively refined patterns and ultimately instruct the Hox genes that then instruct the genes that define the identity of particular segments.